Okay, these are your chapter 14 notes on Mendelian genetics. This is actually going to be split up into two videos. The first video is going to cover stuff before Christmas break, and the second video will cover after. Go through and read page 262 and to 264 to understand the process of how Mendel came to understand some basic principles of heredity. I've left you some space on your notes so that you can fill in some information you find. This reads kind of like a history textbook. Gives you some background information about Mendel and what spurred his interest and what the different things that he did were. So go ahead and just pause the video, go through, do a little reading, take some notes, and then come back. Okay, welcome back. So here's some just basic information. Mendel studied pea plants, which you read, with very simple traits. True breeding means over many generations of self-pollination, the offspring always looked like the parents. You'll hear these terms come up, so I want to make sure you understand them. Hybridization is a mating or a crossing of two true breeding parents. The pea generation is considered the parental generation, and that's two true breeding parents. The F1 generation is the first phalal, which is a hybrid of the offspring of the pea generation. And the F2 would be crossing the F1 generation in plants. So this would be your pea generation here. You have a purple flower and you have a white flower, and you have crossed them, and you now have all purple flowers. And you self-pollinate those. So you're using two of the F1 generation by just using the same plant and pollinating itself. And you get an F2. And what he found is when he mated purple and white flowers, he got all purple, but then when he mated their self-pollinated those purple flowers in the F1 generation, all of a sudden he had about 75% purple, but then he had this other group of white flowers. And he's like, well, where is this coming from? Because I mated purple flowers together, so why would the offspring be white? And that led him to understand the idea of dominance and recessive. He noticed a 3-to-1 ratio. He noticed a 3-to-1 ratio consistently in the F2 generation. He figured out that alternative versions of a gene account for these variations. We now call these alleles. For each character, an organism inherits 2 copies of a gene, one from each parent. If 2 alleles at a locus differ, then one, the dominant allele, determines the organism's appearance. The other, the recessive allele, has no noticeable effect on the organism's appearance. So without really understanding DNA and how genes are inherited by meiosis, he came to this conclusion that this must be what's going on in order to get these traits that skip generations. And it's really very true today, what he figured out. We just know a lot more than he did. And he was working with pea plants, which just happen to have a lot of very simple traits, which is not the case with humans. The law of segregation, the 2 alleles for heritable characteristics segregate or separate independently during gamete formation and end up in different gametes. An egg or a sperm only gets one of the 2 alleles present in the somatic cells. So this is showing that. So I've got a chromosome here. Remember that this is a sister chromatid and this is the other sister chromatid, and they both have the same exact allele because they're exact copies of each other. This one, on the other hand, has a different version of this gene. In this case, this one has the dominant G and this one has the recessive little g. And in this formation of these gametes, this sex cell got a big G, and this one got a little g, and this one got a little g, or sorry, a big G, and this one got a little g, and this one got a little g. And so these separate independently of each other. So when you're talking about multiple alleles, they will not necessarily always go together. If you had this little X here, this little yellow one had a big B on it, and this one had a little b, and this one got the big B, right, and this one got the little b, but that is not necessarily always the case, because this yellow chromosome could have just as easily gone over here with this other yellow chromosome. And so you could have ended up with big G, little b, big G, little b, little g, big B, little g, big B, because those chromosomes separate during meiosis. Now, what we're talking about here is the law of segregation, just saying that the alleles separate from each other. The law of independent assortment, which comes a little later, talks about how they separate, but they also separate independently. And so these combinations don't necessarily aren't always the same. For example, you don't always get brown hair and brown eyes. You can get brown hair and blue eyes, or brown hair and green eyes, if our traits were quite that simple. And this shows that because they separate, we can put them into a Punnett square as the possibilities, and we can predict offspring from there. A Punnett square is a device for predicting the allele composition of offspring from a cross between individuals of known genetic makeup. So say that we know this parent is big B little b, and this parent is big B little b. We're calling this the female and not the male up there. We can actually use the Punnett square to predict what the offspring will look like, as long as we know the genetic makeup already. And you probably remember those from Bio 1. So here's some modern genetic vocabulary. If you don't know the vocabulary, it makes it really, really hard to solve a genetics problem. Most people understand the mathematical concept of solving a Punnett square, but they struggle with identifying what it's asking in the question because they don't understand the vocabulary. So that's my challenge, is make sure you know this vocabulary. So homozygous is an organism that has a pair of identical alleles for a character. So they could have 2 capitals or 2 lower cases, 2 dominant or 2 recessive. Heterozygous has 2 different alleles for a gene. So they have 1 copy of the dominant and 1 copy of the recessive. The phenotype is the organism's appearance, what they physically have. So these would be the words, like, is the plant red or is it white? The genotype is the genetic makeup. That's going to be what 2 alleles are they carrying. Are they carrying a homozygous dominant, a heterozygous, or homozygous recessive? A test cross is something that is used to determine the genotype if we don't know it already. So let's pretend for a moment that we have a mystery plant with purple flowers, and these plants come in either white or purple. We don't know if this purple flower is this or this. We just know purple is dominant and this plant is purple, so it has 2 possible genotypes. Okay, so to determine what the actual genotype is, we can do a test cross with a completely recessive flower, so a white flower. So for example, if the plant we're looking at is homozygous dominant and we test cross it with a recessive, all of our plants should be what? It should be purple, right? All right. Well, if we take this plant and it's heterozygous and we test cross it with a homozygous recessive, then we should expect what outcome? About 50% of them should be white. So if we do this, we know based on this test cross if the plant in question, the mystery plant, is either homozygous dominant or heterozygous. We know what color it is, but figuring out the genotype is the purpose of a test cross. And this works really well if you're dealing with one particular gene at one locus that is only has 2 versions, dominant and recessive, and you only have a purple and a white, or a fluffy and a slimy, I don't know, but it only has 2 versions of the allele. A test cross is when a geneticist breeds an organism of unknown genotype with a recessive homozygote because it can reveal the genotype of that organism. That's what I just walked you through. There are 2 types of crosses we're going to deal with in here. Primarily, we will get into some trihybrids too, but a monohybrid cross is a cross between 2 genotypes following only 1 trait, and a dihybrid crosses 2 genotypes following 2 traits. So this one over here is a monohybrid, and this one here is a dihybrid. And in a monohybrid, we're just looking at, it looks like here we're looking at color. So we've got yellow or green. And so we're just looking at that trait for color. Over here, we are also looking at whether or not the seeds are round or wrinkled. So we're looking at 2 traits at one time. And so we do a double or a dihybrid cross, where we're looking at both traits at once. And these can get really confusing because you can do a trihybrid and you can even go further, and the charts just get bigger and bigger. I'm going to teach you a simpler way to do it today. The law of independent assortment was what I was describing earlier when I'd also described the law of segregation, and it states that each pair of alleles segregates independently of each other, or the other pair of alleles, during gamete formation. In other words, when doing crossing, big Y, big Y, big R, big R, and little y, little r, little r, the Y's and the R's are separate events, and this only applies to genes located on different chromosomes. So this is what I was explaining here. Brown eyes and black hair and blue eyes and red hair. But if these two had been on opposite sides and not all orange on one side and all blue on that side, then you could have gotten brown hair with red eyes and blue eyes with black hair. And just remember that these are very simple examples, because the truth in humans is that eye color and hair color are not single gene traits where you're just dealing with two different versions, brown and red or black and blue or something like that. Okay, so let's do a couple of monohybrid practice problems, just to kind of refresh us from Bio 1. You should all be familiar with this. So we're dealing with flower position of axial and terminal. So if we cross a homozygous dominant axial flower with a heterozygous axial flower, what are we going to get? So we just set up our Punnett square like this. Okay, and we end up with half of them being homozygous dominant and half of them being heterozygous. Well, we know that because dominant is dominant and that's what you see, then that means that 100% of these, or 4 out of 4, are going to be axial in their phenotype. So let's do seed shape and wrinkle. So here we have, oh, sorry, you could do this practice problem off to the side. Go ahead and try that one. I skipped one. Set up the Punnett square. Remember, one parent goes on top and one goes on the side. You should have ended up with a fourth of them being like this, half of them like this, and another fourth being like this. That also means that this is the genotype, but phenotypically, you're going to have 3/4 of them, these guys, being axial, and 1/4 being terminal. So let's do it with seed shape, round and wrinkled. So I'm going to do this one. Well, it looks like 100% of them are going to be big R, little r, which is round. Okay, you try this one. This ends up just kind of like this one up here, except we're using a different trait. A fourth of them are going to be big R, a half of them are going to be big R, little r, and a fourth of them are going to be little r, which means that on phenotype, 3/4 of them are going to be round, and 1/4 of them are going to be wrinkled. And it's interesting, because anytime you have two heterozygotes in one trait, in a Punnett square, a monohybrid cross, you're going to have the same outcome. And you're going to have to memorize these outcomes, because that becomes important in doing larger dihybrid crosses. So in this one here, we have axial seed shape and flower position. So these are actually independent events, but we can do them in one really large monohybrid cross. And I wonder if I have, oh, I don't have a lot of space here. So we'll try to do it right here, and you can follow along with me. So to do this, it's kind of like the FOIL property in math. The big A goes with the big R, and then the big A goes with the big R, and then the little a goes with the big R, and then the little a goes with big R. And then over on this side, we're going to do the same FOIL properties, so big A, big R, big A, little r, little a, big R, little a, little r. And then we're going to do a giant cross. And then you're going to fill in the boxes. Big A, big A, big R, big R. Big A, big A, big R, big R. big A, big R, big R. And you get the idea here. Just filling them in. Oops, that was 2 little A's. Okay, so then you can count these up, and you can actually say, okay, big A, big R, and then count the number that you have over. Now instead of 4, we have 16 boxes, and then big A, big R, little r, and count the number of 16. And I'll let you go ahead and finish that out. And it's kind of obnoxious to do them that way, honestly. It takes a lot of time, and gets kind of repetitive. And then you have to count boxes and hope you don't miss any, and hope your total number adds up to 16 boxes. Go ahead and finish that. You can pause me for a minute. All right, welcome back. So after having done that dihybrid cross and actually having to count boxes and foil these letters, and it gets kind of obnoxious. So I'm going to teach you a faster way. But in order to do that, we have to first memorize some simple monohybrid cross results. So you have to know these, and there's no way around it. You just have to be able to quickly spit them out. So knowing that 2 homozygous dominant always gives you homozygous dominant. Homozygous dominant and a heterozygous gives you half homozygous dominant and half heterozygous. Write these down. Homozygous dominant and homozygous recessive always gives you heterozygous. 2 heterozygous gives you a 4th homozygous dominant, a half heterozygous, and a 4th homozygous recessive. A heterozygous and a homozygous recessive gives you half heterozygous and 1/2 homozygous recessive. And 2 homozygous recessives always gives you homozygous recessive. Also be able to figure out the phenotypic probability of these. So this is always going to give you 100% dominant phenotype. This is always going to give you 100% dominant phenotype, right? It's going to give you the dominant. And we could use any letter here. We don't have to use R. We could use A, we could use B. The letter just usually correlates to the trait we're talking about, and here we're just generalizing dominant versus recessive. It doesn't really matter what letter you use. Here we've got 3/4 that are going to be dominant phenotype, and 1/4 that's going to be recessive phenotype. And here we have half going to be dominant and half recessive. And here we have all recessive phenotypes. So make sure you can do that as well. The laws of probability tell us that we can actually multiply one event by the probability of another event. So for example, the probability of a coin landing heads up twice can be 1/2 each time. So 1/2 times 1/2 is 1/4. And so we can look at, since our alleles separate independently of each other, we can look at the probability of the R allele and the A allele as 2 separate events and then just multiply them together. So the probability of a heterozygous female egg getting the little r allele is half, and the probability of the heterozygous male sperm getting the little r is half. So the probability of them coming together is 1/4. It's the same as doing a Punnett square. So you know that these two come together, and 1/4 of them are going to be little r, and it's the same thing. We're just multiplying those 2 independent events together, the egg and the sperm. And I'll show you how to apply that to matings too. The addition rule: the probability that any one of 2 or more mutually exclusive events will occur is calculated. So mutually exclusive events, it's calculated by adding their individual properties. To determine the probability that the F2 generation will be heterozygous, if the parents are both heterozygous, then we know that the offspring, big R, little r, can get the big R from either mom or dad, but not both, and the same applies to little r. So the probability is 1/4, you're going to get heterozygous, and 1/4 heterozygous, and you add those together and you get a half a possibility of R. So think about that Punnett square between here and here. You are going to get a 1/4 of a chance of getting this combination, here, and the 1/4 of this combination. So if you add those 2/4 together, you get a half probability. The hardest thing for students to understand is when to use the multiplication rule and when to use the addition rule. So we'll do lots of practice with that. And this is just showing the segregation of alleles into eggs and then sperm. So basically, when you're doing a Punnett square, you're showing the probability of getting this from an egg, and this from an egg, this from a sperm, and this from a sperm. And then you're combining those probabilities into the Punnett square to get that the outcome of the offspring. So let's do a few examples. So one of the things I mentioned is that vocabulary is really important. So I've got round and wrinkled and green and yellow. So a homozygous round, yellow. So if it's homozygous, that means it's going to be 2 capitals because it's round and that's dominant, and yellow, which is recessive. So we're going to have homozygous round, yellow. So yellow would be like this. That's the only way to get yellow because it's recessive. And then I have a wrinkled. The only way to get wrinkled is to have 2 of those. And a homozygous green. So that would be like this. If they'd said heterozygous, it would be big G, little g. So there's my answer. And we're going to do a Punnett square with these. And then we'll do the tall and short one too. So these are independent events. So we can look at this and this separately of this and this. So we've got this one by this one. So we know here that there is a probability of getting, because we memorize these, remember, half and half. Okay, and then if we do the G's, we have only this, right? 4/4. So now what we want to know is we want to know when they come together. So what are our options? So this is kind of like there's a property too where you do these diagrams. I don't know if it reminds you of those in math, where you combine this one with this one and this one with this one and see what you get. So we've got this, and then we've got this. These are our 2 genotypic outcomes that we could get. So now we're going to multiply them together. Now, 4/4 is the same thing as saying 1 or saying 2/2. So if we multiply 1/2 by 1, we get a half, and 1/2 by 1, we get a half. So we've now eliminated that huge dihybrid cross, and we've figured out what our possibilities are. Now, for fun, if you want to go back and check my work and do a dihybrid cross with all those 16 boxes and count them up, feel free. Okay. And then this is the genotypic and ratios right here. The phenotypic ratios, we have to look at what the phenotypes would be. So we'd have round and big G. So we'd have round and, what was that? Was G green? Yeah, green. So we have round and green. Nope, not round and round. Round and green. And that would be half. And then we have here wrinkled and green. And that's half. So let's try it with another one. The chances of getting this offspring from this mating. So we've got tall and short, and we're going to go back with the green or yellow. Okay. Oh, and our rounds in there too. So we now have 3 things going on. We could do a big, huge tri-hybrid cross, but that's kind of annoying. So let's look at the T's first. All of our offspring are going to be like this, right? So that's a 1, and we're going to multiply that by the R. So let's look at the R. I put this in the wrong order here. And that, we know that that's a half chance of, or wait, sorry, these are the 2 we're mating. That's a half chance. We know heterozygous by homozygous recessive is going to give you a half chance of getting homozygous recessive. How do we know that? Because we should have memorized that. So we're going to put that down, and then we're going to multiply that by the G's. So here's our parents, and we're looking for this. And so we know that that's also going to be a half chance. So when we multiply all these together, we end up with a 4th chance that we are going to get this offspring. That was a whole lot faster than trying to do a Punnett square and then look for that one in the Punnett square, because that would be a tri-hybrid. That would be more than 16 boxes. Okay, once we get past that, the other thing we're going to be talking about this semester is when we get beyond Mendel. So Mendel looked at pea plants, looked at very simple traits, but that's not always the case. Sometimes traits are more complex. Sometimes there's not just one version or 2 different versions, red and white. Sometimes there's other things going on. So we're going to cover a couple of them in this particular part, and then we'll cover most of them after break. So complete dominance is the phenotype of the heterozygous is the same as the homozygous dominant. That's what we've been talking about so far. So this phenotypically is going to be the same as that. Incomplete dominance, though, is when neither allele is completely dominant. The heterozygous is somewhere in between the two parents. So, for example, in red snapdragons. So that means this is not equal to this. This is going to be something different, and it's a kind of a blending of the two. And so when you mate red and white, you end up with pink instead of red or white. And we write these a little different. We usually say color is our letter, and then we have red, and we have color, and we have white, so that when they come together, when we have this, that's going to equal our incomplete dominant pink. Another example would be codominance. That was when two alleles each affect the phenotype. So this example is in roan cattle. And so these cattle are not red, they're not white, and they're not incomplete dominant, so they're not pink. They're not a blending. They actually have red hairs and they have white hairs. So they have a mix of the two in the phenotype. So they have red and white, not a blending like incomplete. They're codominant. That means red and white are both dominant, so both are seen in the phenotype. And we are going to stop there for this section, and we'll cover more of this later in the year.